Hello everyone, my name is Niels Förtek from Nanion, and on behalf of Nanion it's my great pleasure to welcome all attendees to our webinar on the CardioExcyte. So welcome to everyone from Europe, from Asia, from the US, and also the early birds from the West Coast. Special welcome to our speakers today from Axiogenesis, Herbert Bohlen and Ralf Kettenhofen, as well as from Roche, Liudmila Polounchuk, and from Nanion, my colleague Nadine Becker, who will be giving a brief introduction and will also present on the state of the art of the CardioExcyte. And I would like to point out that you should feel free to ask questions during the whole webinar. Please make sure to use the chat function. And using chat, please ask questions to all panelists. We will take care to answer every single question. We will do so during the webinar as we go along. We will also collect all questions and have one general Q&A session at the end after all three presentations. And whatever questions we cannot answer within the webinar, we will do that offline and follow up with everyone. I think we should get started, and I'd like to hand over for the introduction of Nadine.
Thank you, Niels. Welcome also everyone from my side. Before handing over to our guest speakers from Axiogenesis and Roche, I'm going to give you a brief introduction on the instrument, just briefly focusing on the features and the applications. Okay, so the key features of the CardioExcyte are that it can record impedance and extracellular field potentials at the same time. It does so in a 96-well format with 96 parallel sensors and at very high temporal resolution. It's an ideal tool for high-throughput safety pharmacology, and it has the added features of an environmental control chamber, but can also be used in an incubator. In addition, it's capable of cardiac pacing. We use these 96 well plate sensor plates, showing the electrode setup of a single well over here, to record the impedance and the EFP signals in a consecutive mode. We have an online analysis program set up that is coming with the instrument, and the online analysis is based on adaptive peak detection and triggering. And it does a real-time geometric beat analysis and comes with a comprehensive compound response curve providing IC and EC50 values as well as well-to-well statistics. While this instrument is designed for cardiac applications, it can also, by using the base impedance as the main readout, be used for cell proliferation assays and as a cell quality control device or for toxicity studies. And with this brief introduction, I would like to hand over to our speakers from Axiogenesis. At first, the CEO of Axiogenesis, Herbert Bohlen, will give a brief introduction on the collaborative effort between Nanion and Axiogenesis. And then we will hear about their experience with the CardioExcyte from Ralf Kettenhofen. Over to you, Herbert.
Yeah, thank you, and welcome everybody. This is Herbert Bohlen from Axiogenesis. Ralf is now starting up the presentation. Yeah, here it comes. Okay, so we were very lucky to have the opportunity to test the CardioExcyte 96 with our cardiomyocyte. And we were lucky to compare that also with other instruments, and we found that the instruments, just to start with, what is a very, very good piece of technology. It's very sound. It's a stable produced instrument, and we were very happy to have that here. Also because of the very small footprint that these instruments do have in a certain incubator. Now we use these setups to look for cardiotoxic events and to look for electrophysiological characterization of our cells, mainly for quality control issues. Now, what you will learn about in this presentation that is later proceeded by my colleague Ralf, is the use of the CardioExcyte system and the software. You will learn something about compounds' effect on our Core4U cardiomyocytes as acute and chronic pharmacology is concerned, and we will show you something about about what we have done in the past by sending out pre-plated Core4U cardiomyocytes on plates and sending them all over in Europe and even to the US. Now, give me the chance to give you a brief information about Axiogenesis. I have founded the company in 2000, and meanwhile we have some 30 people working for Axiogenesis, 25 people working here in Cologne, and the rest in the US. Meanwhile, we do have a global distribution network, and as I already mentioned, we also send out living cells either in T-flasks or pre-seeded in, for example, CardioExcyte plates, to have the customer immediately enabled to work with pre-seeded and pre-qualified plates. Now, what we aim for is physiologically relevant tissue of human and murine origin. We aim for maximum purity as for the cardiac cells, and to have a high lot-to-lot stability. That's why we test each and every lot for their particular behavior in the CardioExcyte system to qualify the lots for optimal function. Now we are meanwhile also going into an idea of having a more complex tissue that is made of cardiomyocytes, cardiac fibroblasts, endothelial cells, and meanwhile we are also introducing some neuronal cells to enter into a situation of physiologically relevant tissue. Last but not least, we are going now into high throughput systems in 384 format and 1500 format. And we enable, or we try to enable, our customers within freedom to operate by a complete licensing strategy. Now, Niels and for the team in Munich, that is what we will ask for the next round. These are skeletal muscles, and we would like to have them also tested in the impedance system. Here on the left side you see a single skeletal muscle cell moving, and on the right hand side there is a fully functional syncytium of cross-striated muscles that are contracting whenever you stimulate them, either electrically or with, in that case, acetylcholine. Now, as I mentioned, we would be happy if we could make them also work in the CardioExcyte system. But meanwhile, I hand over now to my colleague Ralf, who knows everything about the instrument. And thank you for the time.
Yeah, hello everybody. Well, warm welcome from my side as well, and thank you, Nanion, for having the opportunity to present a small part of the data that we have generated already with the CardioExcyte. So my intention here in the webinar is now to show you the ease of use of the system and some few drugs that we have applied, and also some results from the shipment that we have done. So getting started with the system is here the protocol, how we treat the cells for the seeding procedure on the NSP96 plate, fibronectin coating, 30,000 cells per well in 200 microliter. After thawing, we need something like 16 to 19 hours before we do the first medium exchange in the morning, and in the afternoon the second one. Next day, medium exchange twice a day, and on day 3, the first time we use the serum-free medium before we apply compounds. The next day on day 4. In principle, cells are ready on day 3 for the experiments. And here is this movie showing the contraction of the cardiomyocytes on the NSP plate, and in the video you see the impedance signal coming up with the contraction. Okay, that was the video. And starting with the handling of the software. Software is the CardioExcyte control software, and this is the first look. If you open the software, on the left you have a window where what's called the logbook. So every step you apply during the experiment will be logged here. You have also a manual interface where you can put in information that you have. Here will be the raw data coming up for each of the wells. You can make a choice of 816 wells on the right side, a little enlarged. In the center you have a single red one that will appear in very large format. Okay, what you do in the next step. The first step to go for the experiment is that you set up your experiment. So this is the window that appears. We have the general window where you define the kind of measurement that you do. You can choose between impedance or combined measurement of the impedance and external field potentials, the sweep duration that you want to do, the numbers and the repetitions. And you can choose the series measurements with actually compound addition. You can define a separate window. You can save it and open up as you like. Here you see the complete 96 well. We can also choose the wells of interest that you want to go for. This is the first step. Second step will be going for the online analysis. When you tap here on this part of the window, next window looks like this, and you have a choice of parameters that are measured already from the raw data that is recorded immediately online, that you can monitor. Well, I will show you later. So these are typical parameters we usually choose for it, and you can install the automatic window size for each of the wells. Okay, next step is then compound addition. This looks like this. So you have, this window opens up if you hit this button here. You have this view on the left side. You have the overview of the compounds. You have here the concentration window in blue, and here you have the complete plate. This is the 96 well plate, and the compounds are numbered, and you can plan your layout for the compounds, and on the right overlapping the concentrations. So you can define your plate layout before the experiment. So you have choice to make. On the right side you see 12 different layouts for your experiments. So you can nicely plan your experiments beforehand. Okay, then you can choose those for the experiment, and then you can start your measurement actually by typing in the number of your plate, and finally you end up usually it now should go for measuring. This is not done because just a screenshot, sorry for this. But during the measurement, you can switch between EMP and EFP mode to visualize here in the windows, what you will see now. Okay, what does it look like when you do the recording? So this is a part of the window, actually. You see the raw data from the EMP signal in this case. And if you hit this button here, you see the raw trace of the impedance signal here. And on the next one is the beat. The mean beat means that it overlays the first signal, and you can see the averages with the following beat ups. So if you have beat irregularities, you can easily monitor this online. And finally, you have the online analysis, where you also have a list of the parameters that I've chosen before, which you want to already analyze and have graphically monitored during the experiment. Very nice. You have always a complete control over what happens in your plate. So these are some typical recordings that you get afterwards with the online analysis tool. On the left, we have the time range and the development of the base impedance of the signals. So at first we get a rise in the signal after seeding the cells, a slight drop, and then the cells start to connect again. And with each of the media exchange that we get an increase of the impedance of the signal from the cells. And here in blue, you see where the compounds were edited, and we get some change here as well. Same for beat rate. So usually we get a very fast beat rate at first when the cells start to connect. It's not very optimal connected, and then afterwards we get a plateau phase, which you can see here, and then you can start your experiments. So very nice control what happens in your plate as well. Amplitude also comes to a plateau. Also medium exchange always shows up with some changes, but only slight, so we always come back to some plateau values, and then you can start your experiment. Okay, so when you want to do the experiments, in this case we have acute pharmacology for Dofetilide. This is a HERG channel blocker, to all of you who are not aware of it. What does it do in the system? So this is actually the experimental setup. There are wells that were chosen. On the left we have DMSO always applied, and here will the concentrations appear. We'll see later this is just baseline recording from EMP and extracellular field potential. Unfortunately, the timescales are not the same. On the left you see 10 seconds, on the right we have 5 seconds window, but it doesn't matter much. So here's a impedance signal from one well, and here I have the field potential. What does it look like if you apply the compounds? So in this case, we choose quite a huge range of compound concentration from 10 nanomolar to 1 micromolar. And this is after 2 minutes of application of the drugs. And already there you can see in the highest doses that we get an interference with the compound, with the signals that we get. So the biggest size here, 500 nanomolar already readily after 2 minutes induced arrhythmic beating, as you can see here from both recordings. From left you see impedance signal, right field potentials. And after 6 minutes, also lower doses at 100 nanomolar come up with arrhythmic beating, and 500 nanomolar almost disappeared already. Okay, so this is what we can record from HERG blockers usually. The second kind of compound that I want to introduce to you, and some very different experiment, is pentamidine. This is a HERG trafficking blocker. It's very interesting because it's another kind of recording. It appears to have also HERG blocker capacities, but in a different way, because it blocks the trafficking of the protein to the plasma membrane. So the number of HERG channels on the plasma membrane is decreased over time. This is not an acute effect, but it will appear about 10 to 12 hours after application of the drug, in theory. What does happen in an NSE plate? So this is baseline recording without compounds. In this case, I picked out the 10 micromolar concentration of the compound. After 2 hours, no acute effect is detected, as you can see. After 10 hours, we already see the first slight changes of the beat rate, and probably also these impedance signal prolongated, getting wider. And here you can nicely see after 20 hours and 30 hours how much this compound interferes with the heart trafficking. This very nicely reflects what is shown in the literature from this compound. Okay, so you have a nice model to monitor chronic effects. And the last thing I want to show you today is actually what we can do with fresh cells or non-frozen cells on an NSE plate. What we do as a special service for customers. So this was a very first initial experiment, shipment of a pre-plated cells with fresh cardiomyocytes to the US. In this case, it was Nanion US, as you might imagine, the first times when you had the CardioExcyte here. Experimental setup was like this. We seeded the fresh cardiomyocytes right from the production on the E-plates, changed the medium to transportation medium. The shipment was done in specialized boxes, some things that we have optimized here at Axiogenesis. Transportation was within 2 days. When it arrived, the plate arrived in the US. They changed the medium to coffee culture medium, and after 24 hours, the experiment was conducted. And what you see here is already the recorded plate from this experiment. So this baseline recording. So I have a nice 96 well plate with signals in all wells here. And Nanion then conducted a compound experiment with 3 compounds and DMSO controls. And as you can see that we have nice reactions here from nifedipine, for instance, at the highest concentrations. We have arrest what we know already, and also the other compounds work very nice, cis-proteanyl, high frequency induction. So this works pretty good. Yeah, so as you can see, this product is ready to use and works pretty nice with fresh cells. So as soon as you get the plate, you can start to conduct your experiments, and there's no risk for the customer that it does not work, because we can also monitor here at Axiogenesis that the cells attach and work very well. Okay, I just skipped this slide with just an analysis to show, and I will briefly summarize what I've said so far. So I hoped I could show you that the CardioExcyte is a very good system to monitor cardiomyocyte effect, and it's easy to use, either software and the hardware as well. The cardiomyocytes work really good in the CardioExcyte for acute and for chronic pharmacology, and we can deliver pre-plated cardiomyocytes on the NSP plates even to the US very easily, and they work pretty nice. Yeah, so one of the features that we have with our cardiomyocytes is 3 days after plating, the cells are ready for experiment, and so you have no delay of your workload and planning. With this, I want to thank you for your attention, and I'm ready to get some questions.
Thank you very much, Ralf, for a very nice talk and for sharing your data and experiences with the CardioExcyte with us. We have a Q&A session at the very end of all of the talks. So for everyone having questions, please submit them to the panelists or hang on until the end of the session, please. So now I would like to hand over to Liudmila Polounchuk from Roche. She's the head of the electrophysiology lab in the preclinical safety department there. And she has also been recording from the CardioExcyte and a number of compounds. We're very excited to hear your talk, Liudmila.
Very much, Nadine.
Hang on just a second. We're moving over the handling to you. This will take another second. Okay. Now you're the speaker, Liudmila. Go ahead.
Thank you. Good morning for those from My first and a very pleasant experience with using CardioExcyte for safety pharmacology studies to ameliorate a cardiac safety pipeline. And I would like to thank Nanion for giving me this opportunity to work with the equipment and present the data, and also for those speakers who already set it up the stage for me, so I don't have to explain much. I will just start with a short introduction to cardiac safety. And in the clinical studies, the QT interval duration serves as a surrogate marker for drug-induced depot arrhythmias, and the majority of drug induces this type of arrhythmias through the same electrophysiological mechanism via the blockade of a specific potassium repolarizing currents in ventricular cardiomyocytes. In preclinical studies, this can be detected by reading animal ECGs in vivo or measuring action potential, duration in tissue preparation, and the hERG potassium current in the cells. Following this correlation between in vivo, tissue, and cellular level, ECH introduced a regulatory guidance for two core battery assays, hERG test to evaluate the potassium currents in vitro, and the ECG reading in unrestricted telemetrized animals in vivo, which are mandatory for each and every drug candidate before entering into clinical trials. And the next slide will give you just a short overview of the generic safety approach, where you see that the hERG test is already conducted at the early stages and followed up by the non-rodent telemetry. So the current strategy is a very hERG, at least at the in vitro part, is a very hERG centric, and focusing only on one component of such a multifactorial process as cardiac rhythm generation, with more than 20 channels and transporters involved in it, eventually leads to two main problems. You either select compounds based on their hERG activity, which may be mitigated and balanced by compound effects on other ion channels, and maybe not that bad, or the other issue is that you may be missing some drugs that may provoke arrhythmia via hERG-independent mechanism. That's why about 2 years ago, the initiative came up, which was heavily supported by regulatory agencies like FDA and also Life Environmental Institute and academic and industrial environment, to basically develop a new or set up a new paradigm for a cardiac safety assessment. And this initiative is called a Comprehensive In Vitro Proarrhythmia Assay, or CiPA, and it provides a more robust early preclinical assessment of proarrhythmic risk of new drug compounds. And the goal of the CiPA initiative will be accomplished by integrating effects of drugs on human cardiac ion currents in heterological expression system, and then followed by the in silico reconstruction of electrophysiological responses linked to proarrhythmia, and then confirmation of effects in on ion current ventricular electrical activity using human-induced pluripotent stem cell-derived cardiomyocytes. So here you just see the schematic and the background element. And when applied to the strategy, this is basically coming from the one-channel centric strategy based on the safety margin assessment of hERG activity to the therapeutic exposure, to a different way of looking at the data, where you really integrate all the information and have tools and technologies and models to integrate the information, and coming up with the multiparameter integrative index or proarrhythmia score. So how you get this multiparameter integrative index? You definitely need new tools to be available and new models to be set it up. And eventually, human-based model, and eventually this would lead to the situation when the thorough QT clinical study can be abandoned from the clinical program. So to start with new functional cardiac safety assessment, when I heard about the new way of looking at the multi-cell preparation, about the hybrid MEA-Impedance technology from Nanion, I thought that may be a way to go. By that time, the stem cell-derived cardiomyocyte became a routine tool already, and then combined with the CardioExcyte, which can read out the both extracellular field potential and impedance beat, may provide you a very interesting combination or unique combination of parameters which are important for cardiac excitation and contractile force. Because extracellular field potential and impedance recording have been already available a while. Actually, we have been trying extracellular field potential since I think a decade, but only combination with impedance that may give you really valuable information about the electromechanical coupling in the cells. So by that time, we also have very good established protocols and experience with Core4U cell from cardiomyocytes, and so far Nanion has been using this already for a while for the system. So we decided to use Core4U cardiomyocytes and plate them on CardioExcyte, and this just presents you very briefly the experimental protocol which we use. So we start with plating the cells and then culturing them for 9 days about in the serum continuum medium. Then the medium is replaced by serum-free medium provided by Axiogenesis as well, and then compounds are added after 12 hours of serum-free medium and incubated with the cells for 24 hours. And while cells are happily sitting in the 96 well plate, the instrument is working and continuously recording. And the next slide shows you actually the results, the outcome of this recording. You see the impedance signal and field potential signals under control condition. This is where you really start to appreciate the versatility and functionality of the software and the equipment itself, but especially software. Because you have a 96 well view, then you have 16 group view where you can focus on certain wells. And then on top of them, you also have a single well view. So you can basically zoom in and out into the experiment. And for everyone working in the early stage, basically knows that the throughput and the flexibility is everything. So then the compound plate is also very easily to set up. I think Ralf already gave you some information on that, and this is just an example of the layout which I used. And initially in the beginning of the trial period, I just tried to do as many wells as possible for one compound, because I didn't know how well does it go. But I would say from my experience, like 4 wells per compound per concentration is more than enough, because the data, the reproducibility is very good, and the results are very consistent. And then after you add, I added the compound actually, and I selected the compounds, just tool compounds, which affect different transporters or receptors on cardiac cells, just to see how different pharmacological agent can modify the response of the cell, to calibrate or to test both the cells and the system. And then this is what you see after the compound has been added, 45 minutes of compound treatment. The interesting point for you to watch is that the well in the lower left corner is the DMSO control, and you see that it has been quite stable over the time, which is a very good sign. For the other wells, of course, it's not that good, and it basically depends on the compound which was added, and it's a good to see also. You can monitor it over longer periods of time. That's also a beauty of the assay, and you can see that sometimes some wells do recover and for others not. But you still can see that the control wells and the signals in the control wells stay stable over 24 hours. So when you have this huge number or amount of data, the challenging task is to integrate the data and prepare them in a summary. And for that, I think also that Nanion developed a very good data analysis toolkit. And here you can see the parameters which you can basically measure online. So you can set it up and you can monitor them already, or do the analysis do post-processing and post-analysis of your data, whatever is better for your study design. And just for simplicity and visualization, for this presentation, I just focused on 6 main parameters, which I consider to be the most relevant to characterize the impedance signal and field potential signals. And these are mainly beating rate for cardiomyocytes, amplitude of both impedance and field potential, and also beat rate regularity index, which give you an indication of is there arrhythmic events, and also pulse width for impedance signal and field potential duration. To visualize these parameters, I mean, of course, you can plot them in a normal graph against the time, but I decided to use the multiplex signatures, and this is how it looks like. So basically, it has multiple axes, and each axis is said to be like 100% response for each parameter, and then you just plot the number which you get already from the data analysis onto that chart. And you can see, for example, that for the HERG blocker, it's like nothing. It's not a secret that you would expect that the field potential duration increases, and also the pulse width of impedance signal increases, amplitude decreases slightly, and also some signs of irregularity appears. And when you look at this chart, you basically already start to have an idea how these parameters are interrelated, and you can also see the dynamic of their change if you just compare the effects after short-term effects and then long-term effects. I just applied it to a few other compounds, and and I would like to share this information with you. For example, also the known IKS blockers. So these are now the slide with more or less potassium channel blockers, and you see here as well that it has a slightly different profile than the HERG blocker. and also the dynamic change of the effects is different. I also had a look at sodium channel blockers. The easiest one is tetrodotoxin. And it's obviously heavily compromised. This is the function of cardiac cells, but slightly recovers after 24 hours. Then flecainide is less effective, let's say, but of course you have to take into consideration the concentration which has been used. Even though I tried to use always the efficacy concentration in the range of like IC50 for a given channel. And this is what you would get after 24 hours of flecainide treatment, for example. Last but not least, calcium channels, which are extremely important for the cardiac safety assessment. And you see that nifedipine as well has a drastic impact on cardiomyocyte function, and it changes slightly. The interesting observation was the treatment with BK, where you see how massively it affects actually everything, beating rate, amplitude, which is understandable. I think it's a kind of expected effect. But it looks like that neither blockade nor the activation of calcium channel is good for cardiomyocyte, because with BK the cells actually were not that happy, and then they only survived up to 6 hours. And I think with that I will stop and wrap up, and in a summary I would like to show basically my conclusion that this system provides sufficient throughput for early cardiac safety assessment. You can easily test up to 24 compounds per plate, and you can even increase the number if you just reduce the number of repeats with the very consistent cell culture protocols. Software package allows you really rapid data handling and real-time analysis. This is really the way to go for the safety screening. And the fast multi-parameter evaluation really allows to integrate parameters which give you basically a good estimate of cardiomyocyte function. And this multiplex signatures maybe is a good tool to highlight cardiac toxicity or efficacy. And on my wish list and looking into the future, I think it would be extremely valuable to work and look at the compound and plate handling, maybe increase the number of plates, because at the moment it's just one plate system. Then of course pacing would help to control the starting conditions. Then overlaying the function of overlaying impedance and field potential signals will provide very valuable information on electromechanical feedback and proarrhythmic potential of drugs, already with some mechanistic insight. And of course, for routine analysis, one would need to establish the user-defined template. Even though the analysis really goes fast, but it would be as end user, I would also prefer to treat all the cells and have standard procedures for my experiments. With that, I would like to thank my direct colleagues, my assistants, and my superiors for supporting this initiative. I would like really cordially thank the Nanion team, which was very helpful with the technical support, and especially Sonja and Ulrich, and also Axiogenesis colleagues. And with that, I would like to thank you for your attention and look forward to the panel discussion.
Okay, thank you very much, Liudmila. That was a really nice talk and very impressive compound profiles. I will now add on to my own talk, and I will be following up with some of our own data and screening efforts, and give some brief technical details as well. And after that, we'll have a common question and answer session where we will try to respond to as many questions as possible. Just a second, we're about to start our presentation. Okay. So again, thank you to all of the speakers so far, and also to the audience who's still hanging on. First of all, I would like to stress why we're so excited about combining impedance and EFP measurements. Liudmila has already mentioned a lot of the aspects. I would just like to repeat that. So the impedance is a perfect measure for contractility, which is important, for example, for seeing inotropic effects. The extracellular field potential, in contrast, is a better measure for electrical conduction. So, for example, measuring the field potential duration to characterize the long QT syndrome. Both of these measures are key to characterize ion channel modulations through pharmacological compounds or in disease models. So together we think that these two measurements make for a really comprehensive and good safety assay setup. So to illustrate this complementary character of the impedance and EFP measurements, I would like to show you an experiment that we did to decouple the contraction and the EFP. What you're seeing on the left-hand side is the impedance signal, and on the right-hand side, the EFP signal. The black trace is our normal baseline impedance signal and EFP signal on the right. And what you see plotted in blue is when we add blebbistatin, which is a highly selective myosin 2 inhibitor. So we can see from the impedance signal that the contraction is completely blocked, while the EFP signal remains basically stable as it is. So if we look at the timeline of this, you can see a clear drop in the contractility, which remains stable as long as blebbistatin is applied, while the EFP signal is largely unaffected. So how does the impedance signal develop over time when we seed the cells? We have already seen some data on this by Ralf, and I would just like to compare a few of the parameters by plotting them on top of each other. So one thing we're following is the base impedance. So once you plate the cells, you get a low base impedance value that is increasing as the cells get more and more attached and flatten out on the sensor plate. And that value will stabilize once the cells have formed a stable monolayer. At the same time, we can follow the amplitude of the beats and the EFP signal and see that they increase as the cells attached and start beating and stabilize as well. Together with those two parameters, monitoring the beat rate, you will see that also the beat rate will stabilize as the base impedance and the amplitude become stable. So this is the point where you would start your experiments, and there are two possible setups that we suggest. One is a cumulative approach where you add increasing concentrations of a drug to each well, so you obtain full dose response curves from the same set of cells. Another option is to do sequential experiments by applying compounds in single in single doses to individual wells, and that will give you the opportunity to monitor long-term effects, just as Liudmila has shown in her talk. So this is one of our pharmacology examples that I would like to show. what I'm showing here is the baseline trace of the raw data of the impedance signal and the EFP signal on the right. And once we add the Dofetilide, we can clearly see early after depolarizations in the impedance signal, and even nicer actually than in the EFP mode. So when we then go on to analyze these data, what we do is to plot a mean beat in the impedance mode and a mean of the extracellular field potential, and here we plotted the means for in a dose-dependent fashion. This was from a cumulative experiment, and in the EFP, what you can see, for example, is a dose-dependent prolongation of the QT interval. So when we look at the dose response curves, I have here selected 4 different parameters to look at. One is the amplitude in red. Then we have the beat regularity index in light blue, the pulse width at half max in middle blue, and in dark blue the beat rate. So from this from the QT prolongation that we pick up in the EFP, we would expect to see a drop in the beat rate, which we do in the impedance signal. And here are the same, a different set of parameters plotted for the EFP signal. And we also see in dark blue a drop in the beat rate, and we can also nicely pick up the prolongation of the field potential duration. Another example for a less selective hERG blocker is quinidine. Again, we have plotted the baseline traces for the impedance and EFP on the top. And again, we see clearly the early after depolarizations in the impedance and the EFP signal. Now, when we look at the mean beat and the EFP signal for the further analysis, again, we see a very clear dose-dependent effect showing a prolongation. And again, we pick this up as an increased prolongation in the mean plots and a drop in the beat rate as would be expected. And we see the same for the impedance signal here. So to look at a compound from a different category, from a different class, we picked nifedipine, an L-type voltage-dependent calcium channel blocker. And this is one example where we can nicely show what we would expect from a drug having a negatively inotropic effect. We see a drop in the impedance amplitude, and in the EFP, in the mean EFP, we can see a dose-dependent reduction in the amplitude as well as the length of the QT interval. And again, this is reflected in the dose response curves. With the reduction of the QT interval, we expect an increase in the beat rate, which is shown here, and also in the EFP data. And we can also nicely see a drop of the amplitude going along with a drop in the pulse width in the impedance data. Now, finally, an example I would like to show for a more complex and more controversial drug, as we take it from the literature, is isoproterenol, a beta-adrenergic agonist. We think that by looking at the impedance signal and the EFP signal, we'll have a better chance to characterize this drug because we'll get a more detailed compound profile. What is somewhat consistent in the literature is that there should be an increase in the beat rate, which we see in both signals, and we can detect a decrease in the amplitude and a slight decrease in the length of the QT interval. So what our efforts are aiming at is to come up with a comprehensive compound library, taking both the impedance measures and not only the 4 parameters that I've shown you here, but also an additional range of analysis parameters, and basically do similar fingerprints as the ones that Liudmila has shown. And as I said, we think that we'll be able to characterize compounds in a lot more detail than as if we were just looking at the impedance or the EFP signals. So what I would like to point out in this preliminary table is that we can clearly make out the hERG blockers. So these are the first 6 or 7. They have a very distinct profile if we look at the field potential duration in our data. And what I would also like to point out that the beat rate are very comparable in the impedance and the EFP signal, as well as the beat regularity index. So we think that we're getting very reliable data, and we have seen by repeating the experiments multiple times that we get highly reproducible data in the impedance as well as in the EFP mode. Okay, I mentioned a few technical features. So at first, we had the CardioExcyte ready to be placed in an incubator for environmental control. In addition, we now have an environmental control chamber, which is shown by the silver lid on the top, so that outside the incubator, we can now control temperature up to 40 degrees, as well as the gas mix and the humidity. And that means we have minimal space requirements for the system. And it's also a very straightforward experimental setup to look at temperature-dependent effects. In addition, we can also do pacing. Here, I'm showing an example of a cell that was spontaneously beating at a frequency of 0.16 Hertz, and we can easily pace it to 1 Hertz, and it will follow for a very long time. Okay, to wrap up, I hope that we could convince you that we're showing you a very comprehensive HTF screening tool for cardiac safety. We can do multi-parameter online analysis, which is straightforward to use. The instrument is suitable for extended applications, for toxicology or cell quality checks beyond cardiomyocytes. We have an environmental control chamber, or the instrument can be used in an incubator, and we are capable of cardiac pacing. With that, I would like to thank our speakers for their great talks. Thank you, Ralf and Liudmila and Heribert. And I would like to encourage you to ask questions. We aim to answer all of your questions either in the Q&A session, or if we don't manage that, we'll respond to you via email. And also, I would like to thank our cell providers who made it possible that we could develop a lot of these features. Thank you all.
All right, also from my side, thanks again to all presenters of today. These are really nice overviews of cell handling, data, and pharmacology. So thanks to you, Ralf and Heribert from Axiogenesis. Also thanks to Liudmila from Roche. And also thanks to you, Nadine, for bringing us up to date on the instrument and its developments. So we have been receiving plenty of questions, and we will be starting with some of those that have appeared more than once. So we have been asked by several people, is it difficult or how easy is it to analyze data with these instruments? You get rather information-rich data, and is data handling, data analysis taking a lot of time? Maybe, I mean, Ralf, you use the instrument and QCing your cells on a routine basis. Maybe that's a question for you. Or maybe you can answer for data analysis.
So, yeah, thank you, Niels, for the question. I'm really pleased to answer this, and I hope I was already able in my presentation to show how easy it is actually. So already on the online analysis tool, you have nice features to monitor the raw data recording, so what's going on, and even the analysis to get an idea of mean beats or your online analysis parameters like beating frequency, the field potential duration, and other parameters that you defined before. You immediately get an idea what happens with your compounds on the cardiac myocytes. So it's very easy.
Well, thanks, Ralf. Then we have a similar question, little different. If you work with unknown compounds, is it possible, when looking at the acquired data, to differentiate for the different compound types or different compound blockers? Maybe Liudmila, you have been looking at different types of compounds. Maybe that's a question for you.
Yes, that's exactly what I did during this pilot study. You can do that if you have kind of calibrated your system, if you have tested already several blockers and you know how the profiles for several blockers or combination look like. Yes, I mean that's the beauty of the integrative analysis, which you can do with this instrument.
Sorry, I wasn't on. So yeah, thank you, Liudmila. I would certainly agree that with these two recording modes, impedance and electric field potential, the idea really is to have complementary data to look at classifying compounds. In this regard, for these two recording modes, we do have another question here. How are you performing hybrid measurements? So this sort of also relates, I think, to what was on your wish list, Liudmila, looking at both recordings and sort of doing overlays. Maybe Nadine, you want to answer how hybrid measurements are done?
Yes, I'm happy to take that question. We have a slide that will help illustrate this. I just need to bring this on. Okay, so I'll start talking while we bring on the slide. We do the measurement consecutively. So we first record the impedance signal and then record the EFP signal. And then there is a small pause between them. And Liudmila, what you were pointing at is can we plot an overlay of the two signals once we have them recorded. We cannot automatically do that yet, but we're aiming to implement a feature to enable us doing this. Sorry, I was hoping to show you the slide.
Yes, thank you, Nadine. I think this would be very helpful. I saw one question maybe regarding the going in the same direction, and just why would you do that? Maybe just to explain a little bit of the background, because as I mentioned, if there is a compound which basically encouples electrical and mechanical conduction, then that's where the cells become very sensitive to and develop arrhythmia. And basically, like, yeah, this is why one would look into this kind of
okay.
Analysis.
So sorry. Now we have the slide illustrating the sequence of the two. And yes, as I said, we're aiming to be able to at least plot an overlay of the signals in the near future. Right now, of course, this can be done manual, but we're looking into finding a less laborious way of doing this. Okay, maybe we go on to the next question.
Yeah, we have a few more here. So some people ask about if you can use only part of a plate for an experiment, that you sort of you only use, say, a column or something like that rather than the whole plate. Maybe Nadine, you can comment on that as well.
Yes, this is possible as long as you keep the plate under sterile conditions. But this is something you will have to do during the entire duration of the experiment anyways. So yes, you can easily use parts of the plate sequentially. Do we have any more questions?
Yeah, we actually have sort of the opposite kind of question. Rather than using only part of a plate, can you also use multiple plates at the same time? So I think this also relates to the wish list that you had, Liudmila, running multiple plates in parallel.
Yes, so you can run multiple plates in parallel because you can save the experimental setups for several plates in the software, and you then switch the plates as you do the recording. Liudmila, I think what you were pointing at was to have them in the instrument at the same time, but this doesn't really seem necessary as you have usually large gaps between individual recording sessions. So I think Ralf mentioned that we can save up to 12 compound plate setups, and this is as many plates as in theory you could do in parallel. I hope that's answering the question.
All right, there is one final question. Are the plates reusable? So this is a question even I can take. A very easy answer from the CEO. Well, you should typically not reuse the plates. Once they are used, you might be able to sort of clean them and use, chemical procedures to clean the sensors and so on. But the sensors will degrade. You will not be able to get as good a signal as with a new plate. The typical mode of operation is single use for the sensor plates. So from looking at the flow of questions, those were the dominant ones. What we will do is we will keep answering all questions that pop up even now. So we will keep answering them for some time. If we don't manage to do it online, we will follow up with all questions that have been asked. So we will send out these via email. So if you have further questions, please type them in or just drop us an email. And I would again very much like to thank our partners at Axiogenesis and Liudmila at Roche for joining us and presenting on their experience with the CardioExcyte. For us at Nanion, it has been a very exciting development, and we're very happy to bring this instrument into the market and then see it being put to use with the stem cell providers and the people in the cardiac safety field. And thanks again to all the attendees for joining us today and your interest in the technology. Please stay tuned. There's more to come. And if there are any open questions, just let us know. With that, we will end our webinar on the CardioExcyte today. And thank to you all again. Take care and bye-bye from Nanion.
Bye-bye.